Good afternoon, ladies and gentlemen. My name is Tien and my partner is Ka. Today we are going to present a paper titled A Multiple Protease Strategy to Optimize the Shotgun Proteomics of Mature Medicinal Cannabis Buds. This paper was written by researchers from Agriculture Victoria Research AgriBio Center in Victoria. It was published in International Journal of Molecular Science with impact factor 2019-2020 of 4.21. Our presentation is divided into 4 parts: introduction, materials and methods, results and discussion, and conclusions. I am going to present the first half and then my friend will continue with the second half of the presentation. Although the genome sequencing has been significantly developed and many studies on the characteristic and synthesis pathway of many compounds in cannabis sativa have been done recently, the proteome of this species is still poorly investigated. Normally, the proteome can be studied by shotgun proteomics, which are divided into bottom-up proteomics and top-down proteomics. In bottom-up proteomics, before analyzed by the mass spectrometry, proteins are digested by the proteolytic enzymes. The flowchart here illustrates the process of 2 different approaches of bottom-up proteomics, which are gel-included and gel-free approach. Compared to gel-free method, in gel-included method, protein mixture is separated and visualized by 2D electrophoresis before digested into peptides. The peptide mixture is then analyzed by mass spectrometry for further identification by database searching. Here this table presents some common proteases used for protein digestion in bottom-up proteomics. The first enzyme is trypsin, which cleaves specifically at arginine and lysine residues. The second enzyme is an enzyme from Lysobacter enzymogenesis bacteria, Lysi. This protease cleaves at carboxyl terminus of lysine residue. The third enzyme is chymotrypsin. This enzyme is less specific than trypsin, and it preferably cleaves large hydrophobic residues such as tyrosine, phenylalanine, and tryptophan, and to a lesser extent, leucine and methionine. Another common protease is an enzyme from Staphylococcus aureus, glucine, and it is a highly specific protease, particularly at acidic conditions. This enzyme cleaves at C-terminal site of glutamic acid residues, while at alkali conditions, it additionally cleaves at C-terminal site of aspartic acid residue. This protease can be used in single, but it has been shown that proteases with different specificity, when used in combination with each other, can improve the coverage of amino acid sequence and the level of proteome exploration. Top-down proteomics allows a full characterization of intact proteins, including proteins shaped by genetic variation, alternative splicing, and post-translational modifications from complex biological systems. The first step of this approach is the separation of the protein mixture, followed by the fragmentation of the proteins for the tandem mass spectrometry. The protein fragments are then identified from generated mass spectra using database searching. Generally, trypsin-based bottom-up proteomics use short peptides from 0.7 to 3 kilodalton, while top-down proteomics typically explore intact protein of 10 to 30 kilodalton. This means that proteins of 3 to 10 kilodalton are not covered by these 2 methods. Nevertheless, this gap can be filled in by middle-down proteomics, which use glucine to yield fewer and larger polypeptides spanning from 3 to 10 kilodalton. Therefore, in this research, based on their reverse experience, the researcher developed a shotgun proteomic to fulfill bottom-up proteomics and middle-down proteomic potentials using combinatorial multiple protease in cannabis buds. Next, I am going to present materials and methods. The experiment was performed in bovine serum albumin and samples of cannabis buds. PSA was used as a positive control for both enzymatic digestion and NLC-MS analysis. Samples were taken from 3 different plants denoted as biological replicate, and each plant sample was done twice. Reference protein PSA and sample powders were extracted following the summarized process in the picture. 3 orthogonal proteases, including chymotrypsin, a ready-to-use mixture of trypsin, lysine, and glucine, were carefully chosen based on their specificity to use peptides ranging from 0.5 to 10 kilodalton as to cover both bottom-up proteomics and middle-down proteomics range. The digestion efficiency of the protease was first tested on their own or in sequence using the reference protein BSA and then applied to the plant sample. And LC-MS was used to analyze single, double, and triple enzyme digest. Proteins are then identified and annotated by the Proteome Discoverer 1.4 using sequence algorithm. Finally, data processing and statistical analysis were performed in Gene Data Expressionist 12.0.
Hi everyone, my name is Ka. I'm the second presenter. In this part of the talk, I will go into the details of the research outcomes and discuss their implications. First of all, we will look at shotgun proteomics of BSA. Here's a table showing the number of MS peaks, MS spectra, and the spectra annotated by sequence program. In general, all LC-MS patterns are complex with a huge number of ions. These figures vary between 77,000 to 100,000 peaks across all replicas. Out of 11 protease preparations, sample treated with glucine followed by chymotrypsin, highlighted in the red box, gives the least number of MS peaks, whereas sample treated with mixture of glucine and chymotrypsin, highlighted in blue, gives the highest number of MS peaks. With MS spectra, they only account for 10% of all the MS peaks on average. After MS spectra has been acquired, data will search again BSA sequence in a database by sequence program, and on average, 70% of the MS spectra gives positive hits or matches, which only account for 1.8% of all the MS peaks. On this slide, the picture on the right-hand side is a collection of heat maps showing all the LC-MS patterns for BSA digests, supplemented by a bar chart. Each heat map illustrates the distribution of all the MS peaks with a mass-to-chart ratio between 300 and 1600 as a result of a certain digest. As can be seen, the patterns observed with G to C and GC are the least and the most complex respectively. However, the digestion mixture with most abundant peaks do not necessarily have more meaningful data. Looking at the graph here, the percentages of MS peaks fragmented by MS illustrated in light gray remain quite constant across all digests. On the other hand, annotated spectra in sequence, in other words, successful hits represented in black bars, reach high proportion when trypsin and glycine are used alone or in combination with other proteases. This suggests that BSA is sensitive to trypsin digestion and explains why it is often used as a standard in proteomics. In addition, statistical analyses were performed in order to investigate the activity of trypsin versus chymotrypsin. As can be seen in principal component analysis results in figure B, the clusters 1, 2, and 3 contain sample treated with chymotrypsin, while chymotrypsin is not present in cluster 4. This analysis results confirms the orthogonality of chymotrypsin, which is different from trypsin activity. An important factor to look at when searching a database is the number of miscleavages. A rule of thumb is to set the number of miscleavages to 2 when using trypsin, but in this research, the author deliberately allows the number of miscleavages to be as high as 6. This is because lower numbers of miscleavages improve discrimination between correct and incorrect matches, but it also assumes the digestion is complete. In reverse, high numbers of miscleavages assumes digestion is partial. The next part will summarize the results of proteomics analysis for medicinal cannabis buds. It is not surprising the LC-MS patterns are much more complicated than what we observe in BSA, which makes sense because biological samples contains more protein and other components. The number of MS peaks ranges from 49,000 to 118,000 peaks, but the interesting thing is MS data has more variance across technical replicas than across biological replicas. This is due to the fact that technical replicas, for example, between Rep 1 and Rep 2, were run independently. Then, the MS data was searched again a cannabis sativas database using sequence program. From this table, we can see on average 29% of the MS spectra gives successful database matches, which accounts for 2.7% of all TMS peaks. Then, data will turn into a histogram similar to the one we saw in BSA. Here in this graph, the proportion of MS peaks fragmented by MS-MS events, illustrated in light gray bars, is fairly constant across all digests. But annotated MS-MS spectra in sequence, illustrated in black, are even more variant than though in the case of BSA. Another thing we can notice is digestion mixture with chymotrypsin alone or in combination provides higher percentages of annotated spectra. For example, DGC mixture had 45% of MS-MS spectra annotated. The author also suggested that in order to improve efficiency of data usage, the sample should be analyzed multiple times and fragmented ions that were already identified should be excluded from later search. Some multivariate analysis were also conducted, and the results show the sample treated with glucose gives the longest peptide, which is larger than 5 kilodalton, and also confirms the orthogonality of proteases chosen in the experiment. Let's recap on what we've spoken about today and give you some take-home message. Due to time limit, we cannot present all the results, but here are the main points that may be useful. First, depending on the research, different proteomics strategies can be selected. For example, top-down proteomics allows the identification of PTMs that cannot be detected with bottom-up approach. The number of miscleavages is a critical parameter because it expands the extent of the search. Majority of MS data has not been exploited, so we need to come up with an optimized method for data usage. This research is a typical example. Lastly, using different digestion enzymes alone or in combination with in shotgun proteomics reveals more information on proteins present in biological samples. This is the end of our presentation. Thank you for listening.